One of the, sort of maybe unsung heroes of AlphaFold was the protein data bank. And it seemed just like this really, prescient initiative that started decades ago where, different types of proteins, it seems like, were very, studiously entered into this database. And then there was a whole, competition yearly around, being able to predict, I guess, novel proteins that had recently been discovered, but maybe were put under some sort of embargo for a period of time. And, that made the work a lot, I would say, more tractable for, the powerful models that, DeepMind was building at the time. I'm curious, as you, look at the, traction that you've had with, for example, predicting affinity, like, is that coming from similar data set? Is there a different data set that's informing that? Like, how does that work?
I would say, largely, as I was mentioning, these models, structure prediction and, the power from, what we're able to learn from, these decades of, biologists, spending this incredible amount of time, understanding the structure of individual complexes is really where a lot of the understanding and representation of these models still come from. Even when we're trying to predict affinity, largely the way that this affinity prediction is done is that it relies on these foundation models that's been trained, largely on the PDB and derivatives of the PDB. And so then on top of that, yes, we are, kind of using all sorts of different kinds of data, and even for affinity and other properties, there has been efforts on trying to collect kind of the data, but the PDB and structure prediction still to this day is sort of like really kind of one of the core ways that we have been able to extract knowledge. Another one that is sometimes kind of less appreciated is the one of the things that actually have enabled, AlphaFold on top of, kind of the PDB and, the rise of machine learning is also the rise of sequencing, and it's interesting because, if you think about, sequence data, that's really not, kind of when people think about AlphaFold, they think about structural data. But in practice, the way that, these models like AlphaFold and Boltz actually operate is by using what's called multiple sequence alignment. So you take a protein sequence and then you go look in basically the proteins that people have sequenced from all sorts of different organisms and so on for similar sequences. You take these sequences, you kind of align them, and you look at where, between different, for example, organisms, they change. And based on those changes, you can actually get some hints about the structure of that protein. And so, sometimes, people, in specific in the field, get somewhat, protective about, as AlphaFold really solve, protein folding because, they leverage multiple sequence alignment. In general, I think, that, we should use all the means that we have to our disposal. But, I think, one of the other, things that really played into the puzzle has been kind of, the rise of sequencing that has allowed us to have, this data from evolution that has, also taught us a lot about the way that, proteins and their structure work.
Well, we're down the street from the Broad Institute, which is famous, I think, for really trying to drive down the cost of sequencing to make it more accessible and in a way to democratize it, to support this kind of research. I'm curious, if you had a magic wand and you wanted to create, the next level of rich data sets that are going to create, better, affinity models that are even more accurate, is it, organizations, governments, labs investing in more sequencing data? Is it another type of data set? Like what comes to mind for you?
Yeah, no, there are definitely a lot of interesting kind of types of data sources that people are trying to generate. I think, largely, as I was saying, we have really relied on this structural data. The problem with this structural data is that, yes, there's been, some improvements, but to some extent it's still basically, one structure at a time. And so people are trying to think about all sorts of, interesting and cool ways of, getting a lot of data, all together. maybe not at the same quality or flexibility that you would have when you generate the structure of an individual protein. And here there are different strategies. For example, some folks, and there is this initiative that the UK government has founded at the Diamond Institute. It's called OpenBind, where they are using this high throughput crystallization techniques to basically predict at the same time the structure, or kind of study at the same time the structure of many individual molecules against a particular protein. And so that is very interesting kind of data source that could potentially give us some amount of data. There is other ways that people are doing kind of generating data with what's called mass spectrometry. So that's a very different, completely different technique, but that's high throughput. And the way that people are using this is sort of like to understand, basically interaction at, sort of like a more coarser grained. For example, can I find, where pairs of proteins are binding? And so that's telling us, about, some of, it's telling us about structure, but it's telling us about structure at a coarse way, but at a much larger scale. And so I think, people are, trying all sorts of cool things, and, I think, we'll try to, use all these different data points, and, what is the right assay that will give the next, kind of, level of improvements, who knows? But it's, the fact that there is so many incredibly smart, bioengineers, trying to figure out, kind of the next assay is kind of like a very cool part about the ecosystem.
I'm curious, so Boltz is a public benefit corporation, as I understand it. Your tools, or at least a lot of them, are open sourced, and you're able to look under the hood and kind of see what the code is and how it's written. I'm curious, when you look at the data set that is informing your tools and allowing to create these kinds of predictions, like, where is that data coming from? Is it from these government data banks and so forth that are freely available? Are you purchasing data sets? Like, what can you share more about that?
Yeah, so I would say, right now, at least, for especially the models that we have open source, most of the data has come from the public domain. And that's a combination of, publications, data that people have shared as part of publications, for example, in the Protein Data Bank, the PDB, as well as, data that comes from patents that people have published. And, in general, the beauty of, biology is that, most of, research in biology is publicly funded. With this public funding also comes a somewhat obligation, more or less, respected to publish kind of all the data that is obtained. And so there is an incredible wealth of data that is out there. Now, it's messy, and so, that's why efforts like the PDB has been so valuable, because they really, force people to, agree on the standard, have certain, checks of quality. And, for other kinds of data where there is not the PDB, we have had to do a lot of that cleaning ourselves, These days, language model can help with that, but it's still a pretty painful process. Now, more recently, we have been, acquiring kind of, a lot of data that we use to train our base models, and this is from all sorts of different sources, but that's really trying to target some of the data that we can't really find in the public domain. And then these are this is kind of the collective data that we use to power kind of our base models, some of which are kind of open source and which are proprietary. And then we also work with companies, and for example, we have announced this partnership with Pfizer, where we combine all that data that we have with the data from these companies, put them together, and obtain a model that sort of like puts take all the data that we can find, and kind of give them hopefully kind of the best model for their particular domain.
I want to dig into that, because I find that fascinating. There's, I think, a sense that inside of these institutes, there are amazing, super valuable data sets that could really advance humanity's understanding of biology. but because of IP issues and otherwise, they're not freely available and they're not freely shared. And I've been curious about like efforts to maybe unlock some of that data. And so every once in a while, I'll hear of like some sort of data consortium between companies that maybe speaks to some sort of data sharing. There's a startup, I believe it's European, called Aferis, if I'm not mistaken, which has this interesting model that seems to be like, we give you this model and we allow it to be trained within these different organizations in a way that somehow protects their IP, which seems maybe promising. I'm not sure, I haven't dug into it further. You're now describing maybe a third path, which is taking this platform and then partnering with a Pfizer in this case and being able to like mesh data sets, but maybe keep that more private to Pfizer. So maybe could you share a little bit more about kind of the strategy that you took and how it compares to those others?
Yeah, so I would say a couple of things. I think, one is that, and maybe we can kind of dig deeper into that, is that, I sometimes, there is, a lot of the discussions in the space, kind of revolves around data and the limitations of data. In practice, I wouldn't say that data has been, the limitation or the unlocking factors, for this space. And maybe we can get into that. But in terms of, when looking at our approach and the data, I think one of the very challenging things is that, you really have to work a lot on the data to make it, integrate into these models. As a machine learning scientist, you end up spending, most of your time, looking at the data and figuring out how to leverage it. And so in general, I'm always, somewhat skeptical of, approaches where, you can't really see the data and, you just think about the data as this, magic, portion that's going to come and is going to improve your model. At the same time, what we are seeing, especially with these companies, is that often, different companies, work on maybe different kinds of targets. And so, within that class of targets that these companies are working on, they have, a lot of, very valuable data that really improves and really moves the needle of the model within those domains. Similarly, when it comes to the chemical space, that, different companies operate on, both on the small molecule side as well as on the biologic side. Different companies really work on, slightly different types of chemical spaces. And these chemical spaces are also somewhat different from, the public one. And so, when you can integrate some data that is, specific to that company, that allows you to, improve the model, to those, particular domains. And so it's not necessarily something that is going to say, it's a magic wand that's going to make the model necessarily better for everyone, but definitely can improve, the model performance on those specific domains where, you are really enriching quite significantly the amount of data, whether it's on the target side or on the candidate therapeutic side.